Immunity or immunopathology is basically garbologic sword. মানে garbologic sword মানে it can do good and bad. দুটোই করতে পারে, ভালো করতে পারে, খারাপও করতে পারে। ভালো কি করে করবে? By ingesting or by killing the antigens. খারাপ কি করে করবে? By giving hypersensitivity reaction or some immunological disease like amyloidosis. Broadly, we can divide it two types: innate immunity, adaptive immunity. Innate immunity also called natural or native immunity. Adaptive also called acquired or specific immunity. Now, innate immunity. Natural or innate immunity, it is the first line of defense. Innate immunity in our body, it is first line of defense. It is, and it reacts immediately. It mediated by cells and molecules that recognize products of microbes, induce rapid protective host reactions. Innate immunity in our body is always present, ready to provide immediate defense against microbes and to eliminate damaged cells. The receptor and component of innate immunity have evolved to serve this purpose. Now the components of innate immunity. Epithelial barriers that block entry of microbes, phagocytic cells, mainly neutrophils, macrophages, dendritic cell, natural killer cell. Among these, the most important is mature dendritic cell. Mature dendritic cell also act as antigen-presenting cell. We will see it later. Now, first is epithelia of the skin and GIT, respiratory tract, monocyte, neutrophil, they act like phagocytes. Monocytes that enter into the tissue are called macrophages. Then dendritic cells are specialized cells presenting epithelia, lymphoid organs, and most of the tissues. They capture protein antigen and display peptide for recognition by T lymphocyte. That's why they are called antigen-presenting cell, APC. Innate lymphoid cell are tissue-resident lymphocytes that lack T-cell antigen receptor and cannot respond to antigen, but instead activated by cytokines and other mediators produced at site of tissue damage. There are some other types of cell as well, like mast cell, which are capable of producing many mediators of inflammation, even epithelial and endothelial cells. Plasma protein, the complement system consists of plasma protein that are activated by microbes. But among that, most important is mature dendritic cell. যখনই ও কোথাও ইনফেকশন হয় বা ওর অ্যান্টিজেন নিতে পায় তখনই কিন্তু সার্ফেসে আসে, তখন ফার্স্ট যায়, মোস্ট কমনলি যায় ম্যাচিওর ডেন্ড্রাইটিক সেল। Now, adaptive immunity. The adaptive immune system consists of lymphocytes and their products, including antibodies. There are two types of adaptive immunity: humoral immunity and cell-mediated immunity. Humoral immunity, which protects against extracellular microbes and their toxins, and it consists of mainly B lymphocyte. And cell-mediated immunity, which is responsible for defense against intracellular microbes, against cancer. Cell-mediated immunity consists of mainly T lymphocyte. It develops more slowly than innate immunity, but is even more powerful. Adaptive immunity is more powerful. Humoral immunity is mediated by B lymphocyte, B for bone marrow derived. And cellular immunity mediated by T lymphocyte, T for thymus derived. cells of adaptive immune system. Mature lymphocytes that have not encountered any antigen for which they are specific and are said to naive. Naive T cell means that T cells which did not expose to antigen. যারা অ্যান্টিজেনে এক্সপোজ হয়নি তাদের বলা হয় naive T cell, naive T lymphocytes. After they are activated by recognition of antigen and other signal, lymphocytes differentiate into effector cell, which perform the function of eliminating microbes. These effector cells. Effector cells, from where effector cells are coming? From T lymphocyte. By T lymphocyte proliferate by effector cell. Very high. Memory cell which live in a stage of heightened awareness and are able to react rapidly and strongly to combat the microbe in the case it recurs. তো কিছু সেল মেমোরি সেল হিসেবে থাকে, যখন ফার্দার যখন আবার অ্যান্টিজেনটা আসবে তখন ও যাতে তাড়াতাড়ি র্যাপিডলি রেসপন্স করতে পারে। দ্যাটস ওয়াই মেমোরি সেলস আর ফর্মড অ্যান্ড দে স্টোরড ইন আওয়ার বডি। সো অ্যান্টিজেন রেকগনিশন বি লিম্ফোসাইটস অ্যান্টিবডি ফর্ম করছে and function neutralization of microbe, phagocytosis, complement activation. And among the T lymphocytes, there are some division between T lymphocytes. One is called helper T lymphocyte, then cytotoxic T lymphocyte or CTL, then regulatory T lymphocyte. And each of the T lymphocytes have different function. Like helper T cell function is activation of macrophages, inflammation, activation and differentiation of T and B lymphocyte. Cytotoxic T lymphocyte, killing of infected cells. Cytotoxic T lymphocyte কি করে না directly kill করে। And regulatory T lymphocyte suppress the immune response. Now, tissues of the immune system. Primary lymphoid organ in which T lymphocyte, B lymphocyte mature and become competent to respond to antigen, and secondary or peripheral lymphoid organ in which adaptive immune response to microbes are initiated. Primary lymphoid organ, secondary lymphoid organ. Primary lymphoid organ means thymus and bone marrow. Secondary lymphoid organs means lymph nodes, spleen, mucosal and cutaneous lymphoid tissues. These are primary and secondary lymphoid organs. Innate immunity, epithelial barriers consist of epithelial barriers, then phagocytes, dendritic cell, complement, natural killer cell. Adaptive immunity consists of B lymphocyte and T lymphocyte. There are some diseases of the immune system. সেই জন্য প্রথমে বললাম it is double-edged sword. For this reason, there are some immunodeficiency disorder by deficient or absent cellular or humoral immune functions, like acquired immunodeficiency syndrome, AIDS. Then hypersensitivity reactions. There are 4 types of hypersensitivity reactions occur in our body. Type 1, type 2, type 3, type 4. Autoimmune disease, when our system fails to recognize self from non-self. The cells which are our own body cells, but system thinks, no, these are not body cells. And that cells kill. Our system, immune system, kills our own cells. That is called autoimmune disease. And possible immune disorder, classical example is amyloidosis. Okay, what is antigen? What is antibody? Antigen defines a substance, usually protein in nature, which when introduced into the tissue stimulate antibody production. Antibody is a protein substance produced as a result of antigen stimulation. There are 5 classes of antibody: IgG, IgA, IgM, IgE, and IgD. Cells of immune system, lymphocyte, monocyte, and macrophage, mast cell, basophil, neutrophil, eosinophil. Lymphocyte. Lymphocyte is the master of human immune system. It is the main component. Our body immune system totally depends on this lymphocyte. Morphologically, lymphocytes appear as a homogeneous group, but functionally two major lymphocyte population, T and B lymphocyte, and there is another cell, NK cell, natural killer cell. B and T lymphocyte proliferate into memory cell, imparting long-lasting immunity against specific antigen, while B cell differentiate into plasma cell. T cell get functionally activated on coming in contact with appropriate antigen. Upon coming in contact with antigen, the macrophage, that is specialized antigen-presenting cell, APC, such as dendritic cell, and the MHC is called major histocompatibility complex. In the macrophage, determine whether invading antigen is to be presented to B cell or T cell. After antigen ঢোকার পরে এরা ডিসাইড করে যে সেলটাকেও কার কাছে নিয়ে যাবে, বি সেলের কাছে না টি সেলের কাছে। That decide by major histocompatibility complex, MHC. There are two types of MHC, MHC class 1 and MHC class 2. Some strong antigen that cannot be killed by antibody response, such as some virus, mycobacteria, tuberculosis, and M. leprae, cancer cell, tissue transplantation antigen, they are presented to T cell. Now B cell. B cell involving humoral immunity by inciting antibody response. B cell comprises 10 to 15% of lymphocyte. On coming in contact with antigen, B cell activated to proliferate plasma cell-like lymphocyte, gain into plasma cell. Common B cell markers CD19, CD20, CD21, CD23. T cell help to provide B cell by T helper cell. Helper T cell help B cell. That's why called T helper cell. It helps B cell to produce immunoglobulins by producing interleukins, IL-4, IL-5, IL-10, IL-13. Now T cell. T cell, cell-mediated immunity and delayed type of hypersensitivity. It comprises of 75 to 80% of lymphocyte, T cell marker CD3, CD7, CD2. Depending upon functional activity, there are two types of T lymphocytes, CD4 or called T helper cell, and CD8, which is called cytotoxic T lymphocyte or T suppressor cells. T helper cell, abbreviated as TH cells. These cells promote and enhance the immune reaction and also termed as T regulatory cells. They carry CD4 molecule on their surface, and that's why called CD4 plus cells. Now the cytokines, TH1, TH2. TH1 elaborate IL-2 and interferon, IFN. TH2 cell elaborate IL-4, IL-5, IL-6, and IL-10. T suppressor cells, called TS cells, they suppress immune reaction but are cytotoxic and actually destroy the invading antigen. That's why called cytotoxic T lymphocyte or CTL. These cells carry CD8 molecules on their surface, that's why called CD8 plus cells. Now natural killer cells. These comprise 10 to 15% of circulating lymphocyte. These lymphocytes do not have B or T cell marker, and neither these cells dependent upon thymus for development, unlike CD4, CD8 cells. NK cell surface molecule, CD2, CD16, CD56. These are markers of natural killer cells. NK cells are part of natural or innate immunity. B cell marker 19, 20, 21, 23. How B cell gives us immunity? By producing plasmacytoid cell. From plasmacytoid cell, plasma cell is producing. And T cell, Th1 cell, Th2 cell, and directly cytotoxic, and NK cell, large granular cell, kill virus infected cells. And ultimately, the cell death occurs by interleukin or interferon. Monocyte macrophage, circulating monocyte and immature macrophages are constitute about 5% of peripheral leukocyte. They remain in circulation for about 3 days before they enter tissue to become macrophages. Now, some important features, functions of macrophages. They recognize the antigen recognition, the most important function of macrophages. They possess cell surface receptor to several extracellular molecules, receptor for cytokine, component of complement, selectin, integrin, and Fc, constant fragment of antibody. The most important function of macrophage is antigen recognition. Then phagocytosis. After recognition of antigen, they will phagocytose that antigen. Then secretory function. Cytokines and prostaglandin. Among cytokines, some of importance are IL-1, IL-2, IL-6, IL-8. These are few important interleukins. Then secretion of protein involving wound healing, like collagenase, elastase, fibroblast growth factor, then acute phase reactants, fibronectin, macroglobulin. Then antigen presentation. When macrophages are unable, first macrophages try to engulf and they want to kill that antigen by itself. But when it is not possible, they can understand now it is not possible for mine to kill the antigen. Then that macrophage will carry that antigen to respective T lymphocyte or B lymphocyte. That will reside by MHC, major histocompatibility complex. Now basophil and mast cell. Basophil and mast cell have IgE surface receptor. Thus on coming in contact with antigen binding to IgE, these cells get activated and release granules, that is, they degranulate. These granules contain histamine, platelet activating factor, heparin. Mast cell and basophil are involved in mediating inflammation in allergic reaction and have a role in wound healing. Now the cytokines. Cytokines is messenger molecule of the immune system. Many functions of leukocytes are stimulated and regulated by secreted proteins called cytokines. Molecularly defined cytokines are called interleukins because they mediate communication between leukocytes. Most cytokines have wide spectrum of effect. Classification: hematopoietin family, GCSF, GM-CSF, erythropoietin, then IL-1 alpha, IL-1 beta, and TNF, platelet-derived growth factor, transforming growth factor, TGF beta family. Cytokines. Then chemokine family, like interleukin 8, monocyte chemokine protein, eotaxin. Mode of action of cytokines. There are 3 modes of action: autocrine, paracrine, endocrine. Cytokines are involved in following action: regulation of growth, inflammatory mediators, like some cytokines are potent mediators of inflammation, they cause inflammation. IL-1, IL-8, TNF-alpha, beta, interferon, gamma. And activation of immune system. The immune system is activated by binding of cytokine to specific cell surface receptor after the cell has interacted with antigen. Then one term is called cytokine storm. Cytokine storm is overstimulation of cytokines. Now major histocompatibility complex, MHC. The function of MHC molecule is to display peptide fragments of protein antigen for recognition by antigen-specific T cells. In humans, it is also called human leukocytic antigen because they were initially found on the surface of leukocyte, like neutrophil. The genes encoding HLA molecules are present on the surface of chromosome number 6. MHC, two types of MHC, MHC class 1 molecule, MHC class 2 molecule. MHC class 1 molecule carries cytotoxic T lymphocyte, CD8 plus T lymphocyte carried by MHC class 1 molecule. And MHC2 antigen, it is carried helper T cell or CD4 plus T cell. CD4 plus T cell carried by MHC2 molecule. And CD8 T lymphocyte carried by MHC class 1 molecule. Now, cell-mediated immunity. Naive T cells are activated by antigen and co-stimulator in peripheral lymphoid organs. They are proliferate and differentiate into effector cells. The principal co-stimulator of T cells are B7 protein. On the surface of the antigen-presenting cell, there is a molecule called co-stimulator. If this co-stimulator not attached, then this reaction will not start. That's why on the antigen-presenting cell, there is a co-stimulator that is called B7 molecule. And it is recognized by CD28 receptor that present on naive T cell. On the surface of naive T cell, there is a molecule that is CD28. And on the surface of antigen-presenting cell, that is called co-stimulator, that is called B7 protein. One of the earliest response of CD4 helper T cell secretion of cytokine IL-2. This creates an occurring loop wherein IL-2 acts as a growth factor that stimulates T cell proliferation and increase in the number of antigen-specific lymphocytes. Now, the number of lymphocytes will increase. The function of helper T cell are mediated by combined action of CD14 ligand and cytokine. So, what happened? The antigen comes and antigen presenting cell. What is antigen presenting cell? Like mature ganglionic cell will go and engulf that antigen. And on the surface of the antigen presenting cell, there will be a molecule that is called co-stimulator B7. And T lymphocyte will go. It will carry to the T lymphocyte by MHC say class 2 molecule. MHC class 2 molecule is a carrying. It just carrying that antigen means antigen with antigen presenting cell. It will carry to the CD4 plus helper T cell. Okay, and then some part will proliferate due to effector T cell. It will proliferate. And IL-2 will secrete. And IL-2 is a growth factor. And the function of helper T cell mediated by combined action of CD40 ligand and cytokines. When CD4 plus helper T cell recognize antigens being displayed by macrophages or B lymphocyte, T cell then express CD40 ligand. which engage CD40 on the macrophages or B cell and activate these cells. Some of the activated CD4 plus T cells differentiate into effector cells that secrete distinct sets of cytokines. Cells of Th1 subset secrete cytokine interferon gamma. This interferon gamma is a macrophage activator. The combination of CD40 and interferon gamma activation results in classical macrophage activation. This CD40 and interferon gamma leading to production of microbicidal substance in macrophages and the destruction of ingested microbes. Activated CD8 T lymphocyte differentiate into cytotoxic T lymphocyte that kills harboring microbes in the cytoplasm. By destroying the infected cells, CTL eliminates the reservoir of infection. CTL also kills tumor cells by recognizing tumor-specific antigen. APC, antigen-presenting cell, and naive CD4 plus T cell, they become combined and produce cytokines. There are three types of cytokines: Th1, Th2, Th17. Each of the cytokines have different functions. Like Th1 produces interferon gamma, it has a function of macrophage activation. Then Th2, it produces IL4, IL5, IL13 that stimulate IgE production. And Th17 that produces IL17 that cause recruitment of neutrophil or monocyte. So each of the cytokines have different functions. So this is the antigen, and this is the antigen presenting cell, mature dendritic cell. It enters in the lymphoid organs. This is the lymphoid organs like lymph node. And here you can see CD4 plus T cell, and they proliferate or called CD4 plus effector cells. If it is not CD4, then it is CD8 cell. Then CD8 T cell is called cytotoxic T lymphocyte. Cytotoxic T lymphocyte directly kills the cells, antigen. And these CD4 plus cells, they differentiate effector and memory cells, migration of effector T cell to the site of antigen, and CD4 plus T effector cells, they produce cytokines. By production of cytokines, macrophage activation occurs, inflammation occurs, and ultimately cell death occurs. Now, humoral immunity. Humoral immunity occurring by activation of B lymphocyte and elimination of extracellular microbes. Upon activation, B lymphocyte proliferate and then differentiate into plasma cell that secrete different classes of antibody. So B lymphocyte produce plasma cytoid cell, again plasma cell, and plasma cell produce some antibody. So, and that antibody, there are some functions of antibody which kills the microbes, like neutralization of microbes and toxins, opsonization and phagocytosis, antibody-dependent cytotoxicity, lysis of microbes, phagocytosis of opsonized microbes. And this is the proliferation, differentiation. It has three functions. First is antibody secretion, then class switching, then affinity maturation. Class switching means one class of antibody means it was IgM plus IgD, but after class switching it becomes IgG. It is called class switching. Now, the immune response to cancer. It has long been known that body's immune system can recognize tumor cell as non-self and they attempt to destroy them and limit the spread of cancer. Certain cancer evoke significant lymphocytic infiltrate composed of immunocompetent cells, and such tumor have somewhat better prognosis, like medullary carcinoma of breast, seminoma testis. So in our body, we have some amount of some sort of defense system against cancer cell. When cancer cell produce in our body, first our body immune tries to kill that cancer cell. Rarely a cancer may spontaneously regress partially or completely, probably under the influence of host defense mechanism. For example, rare spontaneous disappearance of malignant melanoma temporarily from the primary site, which may then reappear as metastasis. Primary site melanoma can disappear হয়ে গেল, but it will reappear some other site, that is called metastasis. It is highly unusual to have primary and secondary tumor in the spleen due to its ability to destroy the growth and proliferation of tumor cells. Existence of immune surveillance is substantiated by increased frequency of cancer in immunodeficient host. In HIV patient, there are incidence of cancer is more. That's because of their immune system is compromised. That's why in HIV positive patient, some sort of cancer occur in more prevalence. Assuming the immune system is capable of recognizing and eliminating nascent cancer, it follows that the tumors that grow out in immunocompetent individual must be composed of cells that are either invisible to the host immune system or that activate mechanism that suppress host immunity. The following session explore some important questions like what is the nature of tumor antigen? What host effector system recognize tumor cells? How do tumor evade this host mechanism? How can immune reaction against tumor be exploited therapeutically? So first question, tumor antigen. What is tumor antigen? Tumor cell express some surface antigen, which have been seen in animal and in some human tumors, like tumor-specific antigen, that is called TSA, and tumor-associated antigen, TAA. However, it is now known that TSA and TAA can both be present on normal cell as well as, and categorization into TSA and TAA does not hold true. Thus, presently distinction of tumor antigen is based on their recognition by the host immune cell, that is CD8 plus T cell or cytotoxic T lymphocyte, and by molecular structure of the tumor antigen. So TSA and TAA, that concept is not holds true nowadays. Nowadays, it is based on CD8 plus T cell, that is called cytotoxic T lymphocyte, and by molecular structure of the tumor antigens. Currently, various groups of tumor antigen are as follows: oncoproteins from mutated oncogenes, products of RAS, BCR, RABL, CD4, protein products of tumor suppressor gene. The examples are mutated proteins of p53 and beta-adenine. Overexpressed cellular proteins, for example, in melanoma, the tumor antigen is structurally normal melanocyte-specific protein, tyrosinase, which is overexpressed compared with normal cell. Similarly, HER2-neu protein is overexpressed in many cases of breast cancer. Then abnormally expressed cellular proteins. Sometimes a cellular protein is present in some normal cell but is abnormally expressed on the surface of tumor cell in some cancer. The classic example is present of MAGE gene. It is silent in normal adult tissue except in male germline, but MAGE gene is expressed on surface of many tumors such as melanoma. That is called melanoma antigen. Cancer of liver, lung, stomach, esophagus. So this gene called cancer of liver, lung, stomach and esophagus. This MAGE gene. Tumor antigen from viral oncoprotein. There are some viral oncoprotein like HPV, human papilloma virus, E6, E7, and cervical cancer and EBNA, Epstein-Barr virus nucleic acid protein, EBNA. This virus expressed in cause EBV in Burkitt's lymphoma. Then second is anti-tumor immune response. This killing of tumor cell is caused by cytotoxic T lymphocyte, CTL, or CD8 plus T lymphocyte. This mechanism predominant because the majority of tumor neoantigen consists of mutated gene product of viral protein that are endogenously synthesized and presented in the context of MHC class 1 molecules, because previously I told MHC class 1 molecule will carry CD8 T lymphocyte or cytotoxic T lymphocyte. Cell-mediated mechanism. This is the main mechanism of destruction of tumor cell by host. The following cellular response can destroy the tumor cell and induce tumor immunity in humans. Specifically sensitized cytotoxic T lymphocyte, that is CD8 plus T cell, are directly cytotoxic to the target cell. Then natural killer cell, NK cell. NK cell acts by antibody-dependent cellular cytotoxicity or ADCC. NK cell together with T lymphocyte are the first line of defense against tumor cell and can lyse tumor cell. It can lyse tumor cell. NK cell with T lymphocyte. Then macrophages. Macrophages are activated by interferon gamma secreted by T cell and NK cell. Therefore, there is close collaboration of these two subpopulations of lymphocyte and macrophage. Now, humoral mechanism. As such, there is no anti-tumor humoral antibody which are effective against cancer cells in vivo. However, in vitro humoral antibody may kill tumor cell, but in vivo there is no humoral mechanism. There is cell-mediated immunity, and mainly by natural killer cell and cytotoxic T lymphocyte, CD8 T lymphocyte. Then third is immune regulatory mechanism. Most cancer grow relentlessly in spite of host immunity. This is explained due to delayed controlling mechanism of immunity, and that during progression of cancer, immunity cell may disappear. Cytotoxic T cell and NK cell may play a self-regulatory role. Immunization mediated by various acquired carcinogenic agents. Then third is cancer immunotherapy. Current status of immunotherapy is focused on following three main approaches. Cellular immunotherapy consists of infusion of tumor-specific cytotoxic T cells. That is called tumor-infiltrating lymphocyte. What is tumor-infiltrating lymphocyte? In a tumor, suppose a tumor present in breast or anywhere else. So around the tumor, there will be some lymphocyte. Lymphocyte infiltration will be present. So the more lymphocytic infiltration present around the tumor cells, the prognosis will be better of that tumor. The tumor prognosis will be better. That is called tumor infiltrating lymphocyte, TIL. More the lymphocyte around the tumor cell, the prognosis will be better. The patient's peripheral blood lymphocytes are cultured with interleukin-2, which generate lymphocyte-activated killer cell having potent anti-tumor effect. Cytokine therapy is used to build up specific and non-specific host defense. These include interleukin-2, interferon alpha, interferon gamma, TNF alpha, and GM-CSF. And monoclonal antibody therapy is undergoing trial against CD20 molecule of B-cell and certain B-cell leukemia and lymphoma. One of the medicine name is rituximab, which is under trial and sometimes it is used, that is against this CD20 molecule, rituximab. And it is given in some B-cell leukemia, B-cell lymphoma patients, it is given rituximab. It is called monoclonal antibody therapy.